Hi everyone, I hope you're doing very well. As you can see, we are in a symposium which is called Women in Neuroscience Symposium 2022. I just want to say it is a pleasure to welcome you to Women in Neuroscience Symposium 2022. And the Women in Neuroscience Symposium will be held on 12 to 14 August 2022, but we're going to have a workshop actually on 15 August. I'm going to mention it later completely and totally, but there will be a workshop too, and which is held by International Center for Neuroscience Research in Georgia. And the symposium will be held simultaneously with the Women's Neuroscience Festival and supported by the International Brain Research Organization. Are the highlights of the symposium: recent advances in basic and clinical research in neuroscience, and gender gap in research, women's insight in neuroscience, and gender diversity and new discoveries in neuroscience. The organizer and chair of this symposium is Dr. Nasrollah Moradikor, Ph.D., Research Director, International Center for Neuroscience Research, Institute for Intelligence Research from Tbilisi, Georgia. I'm just going to ask him to come. Are you ready, Professor?
Yes.
Okay, welcome.
Yes. Thank you, Helen, for the complete explanation. Hello, everyone. Good morning. I'm Nasrollah, organizer of the conference. We are very happy to see you at the largest event on women in neuroscience. Welcome all to Women in Neuroscience 2022. The symposium will be held by International Center for Neuroscience Research in Georgia with support of International Brain Research Organization. At the symposium, the gender gap in research and recent advances in basic and clinical neuroscience research will be discussed by invited and keynote lectures at the interesting panels. I have a special thanks to the scientific and organizing committee, national and international speakers, International Brain Research Organization, European Society for Neurochemistry, Women in Neuroscience UK, Women in Neuroscience Nigeria, Georgia Neuroscience Associations, Ivan Javatishvili Tbilisi State University, Malaysia Neuroscience Society, German Neuroscience Society, Italian Neuroscience Society, and Science Team Institute, and other organizations that supported and helped us to organizing the symposium. I hope you enjoy during the symposium by famous neuroscientists and interesting panels. Thank you so much for your participation. Thank you.
Professor Dr. Shartley Ibidiadan, Ph.D., Neuroinflammation Group, Immunology Unit, Department of Physiology and Faculty of Medicine and Health Sciences, University Putra, Malaysia.
For the longest time, we assumed that microglia are just monocytes within our peripheral circulation, our systemic circulation, that have entered the brain and have differentiated to become microglia. We just thought that they were monocytes that had entered the brain parenchyma. This is the group, past and present, and I'm obviously very thankful to my fund providers and all my students. I wouldn't be here without them. And once again, thank you very much to the organizers and Dr. Nasrollah. And on a last note, being the Women of Neuroscience Symposium.
Professor Dr. Ilana Gosic, Ph.D., and she is the director of the Elton Laboratory from Faculty of Medicine, Tel Aviv University.
And you can see it here, when we knock down ADNP, we lose processes, we lose the axons, and we can protect the nerve cells. But if we add a very small fragment of ADNP, this is me and my group, and this is my group over the last 40 years. So I mentored many students, many women students, and they all were excellent. And by that I will end my talk. Thank you very much.
Introduce the co-chair, Dr. Natalia Nalibeva, Ph.D. from Leeds University, United Kingdom Institute of Evolutionary Physiology and Biochemistry from St. Petersburg, Russia.
We looked into the mitotic centrosome this time, and you can see that when NLS neurofibromins are missing, they have huge centrosomes, much bigger, that they do not really colocalize with neurofibromin. So the next was to see how asters were formed. People that are doing it, and thank you for your patience with all my sharing problems.
Dr. Lilia Chomova, PhD from Henslow Research Fellow, PhD, Henslow Research Fellow, Director of Studies for Psychological and Behavioral Studies.
We actually know very little about how unmet social needs affect our brain and behavior. So in my research, I'm really interested in studying how does social disconnection affect the brain? In this particular time in our lives, we undergo sort of a transformation of our social world, which means that the center or the focus of our social interactions shifts away from our parents towards peers. While at the same time, our brains continue to develop. And this is something that we see not only in humans, but also in other species. I would like to thank all the people who helped me with these studies and the funding agencies, and also thank you for your attention.
Associate Professor Dr. Aslina Ahmad Anwar, Ph.D. from Department of Biomedical Sciences, Faculty of Medicine, University of Malaya.
I work at the University of Malaya, and my sort of bread and butter research, really, what gets me papers and what my students are mainly working on is on the neurogenetics of neurodegenerative diseases. Studies that have shown that shift workers are also at high risk of getting cancer, and they think that is because of this overexposure to light at an inappropriate time of day, which is the nighttime. So thank you so much.
This panel is Dr. Elizabeth Tbilisi, English, PhD student at the University of Cambridge, United Kingdom.
My co-chair for today, and she's from the events team at Women in Neuroscience UK, and she's a neuroscience undergraduate student as well at Cardiff University. So Shloka, please could you introduce yourself and also introduce our first speaker?
Thank you, Lizzie, for the wonderful introduction. Hope everybody's doing well. I'm Shloka, and I'm an undergraduate student in neuroscience at Cardiff University, UK. It is my absolute pleasure to be introducing our first speaker for today. She is Victoria, and she is a PhD student at King's College London, and she's going to be talking to us about barriers to autism diagnosis for autistic women and girls. Over to you, Victoria.
So what we found was that in the diagnosed group, the women had significantly higher scores than the men across all three measures. So that's looking then at the blue line and the dark green line being the female, across the CAPQ, which is a self-report measure, the Camo Theory of Mind measure, and the Camo SRS, which is the self-reported measure.
Here with Pink Concussions, which is an organisation that was founded by Catherine in the US, and she's been a really inspirational brain injury advocate for the last 10 years. And more recently, Mary Leflergue in Canada and myself in the UK have come on board to try and extend the reach of Pink's advocacy.
What is Pink Concussions? So we are a nonprofit formed in 2015, and we advocate for better research in the research community. We research TBI differences ourselves. We do trainings for the medical community through Concussions is a blend of research from sports, from accidents, from domestic violence and gender-based violence, and military TBI research. And all those together form the breaking down of the silos together to form the research on brain injury.
Dementia has been the leading cause of death for women in the UK since 2011. So this was even the case during the COVID-19 pandemic. And we found that 2 in 3 people with dementia are women, and that longer life expectancy of women alone does not explain this discrepancy.
I'm hoping that those sexist views are way in the past, and that women can, as I see so many women now in the field. So I'm just excited by seeing all the young female faces.
Dr. Irma Khachideh, PhD from the Department of Behavior and Cognitive Functions and Human Psychophysiology.
Our EEG Hub-G, which is not only service, we use this for scientific data for research, and they are done 2 projects. One is already finished. This is the first, which is aims to study EEG biomarkers in children with different, in children with epilepsy and during their pharmacotherapy, which is already finished. And second one is the ongoing project, which is a study EEG patterns in patients with different CNS disorders, which is an ongoing project. I will represent.
I'm Dr. Tatiana Kaczynska, PhD, Assistant Professor, Human and Animal Physiology Department from Lysia, Ukraine.
I'll talk about Ukrainian nowadays. We have a very difficult political situation, and we have a very dangerous situation. Therefore, level of aggression in our country every day becomes more and more. And we have to help our teenagers, and we have to help observe were observed in light and stimulate isolate room, and each neurofeedback training session took 10 minutes of targeting increase in alpha band power in FZ.
Professor Dr. Sohaila Karimi, PhD, Professor of Physiology and Pathophysiology, University of Manitoba, Canada.
So what's showing is that ISP-ILP peptides, they're basically activating Wnt signaling and overriding the inhibitory effects of CSPG. And also we confirmed that Wnt is important in these neurons because even in the control of CSPG treated neurons in culture, when we inhibited Wnt signaling.
The Assistant Professor of Pharmacology Division, University Institute of Pharmaceuticals.
Most of the studies, this 3 milligram per kg dose is used, which is injected bilaterally in divided dose, means 1.5 milligram and 1.5 milligram per kg in each lateral ventricle. And it may be a single injection, or it may be repeated after 3 days. landmark.
The chair of this panel is Dr. Antonella Catamassa, Division of Pharmacology, Department of Neuroscience, School of Medicine, Federico, University of Naples. From Naples, it's
for me to introduce and to chair this symposium, and thank you for the kind introduction. I think we can start with the first speaker.
High anxiety behavior, but also depressive-like behavior. And what she did first was to use the cells from these hub animals and another animal model of endogenous depression, which is the so-called the brain glucogen supply, the energy reserve, the regulation of extracellular ions. They're also important for the regulation of blood-brain barrier because they're able to uptake water and glucose and release molecules that are involved in the dilation and the contraction of the vessels is included. And of course, I want to also to thank all the people that are the members of my team.
We found a strong increase of N6-1 when we co-cultured our astrocytes with microglia and we exposed them to a GB followed by reoxygenation. In particular, we performed also quantitative analysis, and we found that the number of GFP and N6-1 double positive cells appeared significantly upregulated in our
the level of two markers of astrogliosis. On the same way, we treat our cells, this time with CETA, as we perform in vivo with our mice. This time, the in vitro treatment was performed for 7 days, mimicking a chronic treatment. And again, if we look at GFAP and S100 beta markers, after the treatment with CETA, we managed to obtain a lower level of
going into in what way the phenotype was improved, because we also found that not only did the cells affect the brain they were transplanted into, but also the brain affected the cells that were transplanted. And we found this was actually a very interesting interaction, so we wanted to look into that.
Professor Dr. Ji-Hyun Kim, Ph.D. from Deakin University, Impact Institute for Mental and Physical Health and Clinical Translation.
Ages and genders, but how that memory changes. So in extinction, we repeatedly present the stimulus without any outcome. So you keep riding your car, but there's no accident. And you keep drinking that brand, but then you don't feel good anymore.
Or you don't. I'm just going to introduce Dr. Sarnas Tanga, Ph.D. Development and Disease, Neuroscience Institute, Cavalieri Ottolenghi.
Today we are in this building, so our laboratories are in the Institute of Neuroscience Cavalieri Ottolenghi, which is this building outside from Turin. The 70% of researchers working in this institute are women. I thank all the team of our group that is here for everyday life.
So Dr. Kamai Esrai, PhD, Professor and Coordinator, Division of Physiology.
Showing that if there is an increase in cerebral crus area, it could be an indicator of treatment-resistant depression that was observed in certain studies.
Dr. Marianne Norzian, Professor of Neurology, Director, Cognitive Neurology and Neuropsychiatry Division, Department of Psychiatry.
Less education has a very important role in dementia risk reduction. If you remove this risk factor, less education and illiteracy, you can remove 7% of dementia prevalence.
And the chair of this panel is dear Dr. Sandra Diaz, Simulation and Data Lab Neuroscience from Gilead Supercomputing Center.
We are now ready. We can start with Professor Pasha Kanawada. She is joining us today. She's Professor of Computational Neuroanatomy at the University of Cologne.
Models, they serve as platforms for testing theories of cortical dynamics and function. And this is the idea is that there's currently a vast zoo of available models, each of which explains perhaps one or a few phenomena. But the name of this research infrastructure is eBrains, and this is just showing you the web portal here, which is the entry point to eBrains. There's different tools are part of eBrains. This is just summarizing a couple of them. There are simulators that are allowing different levels of resolution, so very fine-grained cell models, and then more towards point neuron models.
Dr. Tamar Abikachili from British Center of Experimental Biomedicine in order to talk about
exposure. Hippocampal field, especially CA1, show different phases of cellular pathology, predominantly affecting interneurons and synapses. Asymmetrical synapses are sometimes regarded as highly active.
I'm just going to ask Dr. Shina Orich, BBS PhD, anatomy lecturer, University of Port Harcourt, Nigeria.
So this is actually what the women face. We face all manner of mental disorders, depression, neurodegenerative disease, arthritis, cancer, and all that. And coupled with the fact that the funding is not here, the people are financially incapacitated. Because if you have the money, you can go to the market, get whatever you want to get. Fruits and vegetables to mop up the free radicals.
Dr. Yogita Adlaka from Translation National Health Science and Technology Institute, Hyderabad, Haryana in India.
And expressed microRNA 137, and we stained our cells with the mitotracker green. And with the microscopy, we found that the fluorescence intensity was increased in presence of MIR137, which suggests that the mitochondrial number was actually increased.
I'm Sima Papu, PhD candidate of Neuroscience at Chemite and Neuroscience Research.
NMES is a non-invasive technique in which electrodes are placed on targeted muscle groups on the skin, and there are different current intensities in the studies which can be used, and different montages can be used in this technique.
Dr. Nino Mazdiashvili, associate professor at David Tbilisi Medical University at Tbilisi.
Alpha activity frequency changes during sensory motor task and complex sensory motor task frequency, and as well as in case of the power. It was stronger, this inter-individual difference.
Dr. Yusra Al-Hilali from Dementia Research Group, Sussex Neuroscience, School of Life Sciences, University of Sussex, East Sussex from United States.
I was sampled by atomophores microscope, and the data from the 72 filaments show that DJE filaments have about 72.7 nanometer crossover peak, and it's similar to the data collected from the TM in
our workshop. The title of this workshop is actually Mind Your Heart, Introduction to the Electrophysiological and Methodological Aspects of it.
Thank you very much for inviting me to facilitate this workshop. It was an honor, and especially the reason that I've chosen to discuss about the heart-brain connection is because it's an emerging field. We know electrophysiology in terms of electroencephalography. That was all planned. There was several destinations. Usually it doesn't happen when we are recording the data in original settings. That is because we have a dedicated space. But right now, because we needed the internet connection, I have changed the space for data connection, and there are certain interferences from the electrical systems around. We have ground electrode here, and we have the reference electrode over here. Then we connected the leads to the device, which has channel A, which reflects channels from 1 to 4.